Good morning or afternoon to you. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society, and I'm joined by my colleague,
Silvia Crescioli from the Cancer Antibody Discovery and Immunotherapy Group at King's College London.
We will be your moderators today. This is the third in a series of webinars designed to inform and educate our members, as well as the broader scientific community, about antibody discovery and development. The title of today's webinar is Deep Mining of Early Antibody Response in COVID-19 Patients Yields Potent Neutralizers and Reveals a High Level of Convergence. Our speaker is Dr. John Mccafferty, who is the Chief Scientific Officer at IONTAS. Over his long and illustrious career, Dr. Mccafferty has been involved in many aspects of discovery and development of antibody therapeutics, and we are thrilled that he could join us today. Please note, the webinar is being recorded. We will take questions at the end of the presentation, so please add your questions to the Q&A box in the viewer. Without further ado, I now turn the show over to Dr. Mccafferty.
Janice, thank you very much. Thanks for the introduction and thanks for the opportunity to speak and live as well to keep me keen. It's hard to believe actually that just over a year ago nobody had ever heard of SARS-CoV-2 or indeed the COVID-19 the disease it causes. From a UK perspective, actually the first case landed in the country just at the end of January 2020. And to date, there's been over 4 million infections, 126,000 deaths in that short time. And during that year, there's just been such a huge coordinated and collaborative effort with people advancing, every week, every month. There's been unprecedented use of the word unprecedented. And from a UK perspective, from a standing start, we had this virus that nobody knew about, 12, 14 months ago. And to date, we've now got 3 approved vaccines and 25 million people, 50% of the adult population of the UK have already been immunised. So quite a staggering sort of progress and effort during that time. During the first lockdown here in March, I became aware of a Bioindustry Association initiative, a consortium looking to generate neutralizing antibodies. And so I got in touch with Jane Osborne, who was heading up that effort, really putting her hand up to contribute. And so that led to the work that I'm going to describe today, which was carried out really by my colleagues here at IONTAS while carrying on their day job. And I pay tribute to them for the work that they covered in that time, obviously greatly assisted by our collaborators. So for the talk today, just give me a second. So today I'm describing that work. It's actually been published, this Bullen et al. Paper that you can see on the left, and it's currently undergoing peer review for publication in a mainstream journal. So I will go through a little bit of an introduction to the virus and then go on to talk about the donor-derived phage antibody libraries that we created from 18 convalescent donors. On the one hand, I'll talk about the analysis of the donor response, and on the other hand, the goal of this ultimately was to try and generate neutralizing antibodies that might go forward as therapeutics, and I'll go through the work of that. And then finally, the story has sort of changed in recent months with the emergence of these viral variants, and I'll say something about that as well. So this is a cryo-electrotomography image from the publication that you can see here from a few months ago. Actually, the great thing about this is every paper was from a few months ago, or at least a 2020 date. This one from a few months ago is looking at the actual structure of the virus. So it's got a 30 kilobase RNA genome, probably the most intensely studied stretched nucleic acid in history, encodes about 29 proteins with 4 major structural proteins. From a an antibody point of view and from a neutralization point of view, the spike protein is the dominant one. Over 90% of the neutralizing antibodies are there. And this generates or gets into epithelial cells of the lung by binding to ACE2 on the cell surface. And from this study here, the authors estimated a mean of around 26 of these spike proteins per virus. So it's a lot less dense than you might see with influenza virus. And they also talk about a great degree of flexibility of these spike proteins as they move around on the virus. So now looking more closely at that spike protein. So it's basically a protein of just under 1,300 amino acids. There's an N-terminal domain, which itself can be a target for antibodies, but is also quite variable. Shown in orange here is the receptor binding domain that is used to bind to the ACE receptor on cells. Following that RBD domain, there's a stretch with some protease sites, and upon binding to the receptor, we have cleavage there that then unleashes the C-terminal domain of the protein. We have these heptad repeats, HR1 and 2, that together drive the change in conformation and basically allow the insertion of the fusion peptide, FP, into the membrane of the cell. There's also a transmembrane domain shown here, TM, anchoring the protein into the virus and also on the membrane of the production cell. It's also got a very small cytoplasmic domain of about 37 amino acids, which interestingly has got about 8 cysteines in it, and there's another couple of cysteines in the transmembrane domain. Not entirely sure what's driving that, but it is something that is retained in a number of the coronaviruses. The protein itself forms a trimer, and I've shown in bold one of the monomer units on the left, and it adopts very often this so-called closed conformation, which can switch to what's shown on the right, which is referred to as the open conformation. And in this image, we've got one leaf of the trimer opening up. Shown in orange is the same color code, the receptor binding domain that binds ACE2 receptor. And in fact, in yellow, I've highlighted a receptor binding motif that actually makes contact with ACE2. And this site is not really available or so available when it's in the closed conformation. So part of the binding involves this opening of this trimer domain. This is a heavily glycosylated protein, which may be part of the reason why it manages to evade immunity, at least to begin with. So why does it have such a devastating effect on the population? This is again a review from about a month ago, where people have kind of tried to come up with a framework or a model of what's actually going on with SARS-CoV-2 infections. For a sort of more regular, shall we say, normal virus, basically the adaptive immune system deals with it pretty quickly, as represented on the lower panel on the left. The adaptive immune system, shown in green, gets going, prevents viral replication, creates an antiviral environment, and basically triggers the adaptive immune system to proliferate. Of course, there we have individual T cells and B cells that have to multiply, and so on. So the innate system is a generic approach to getting things started, while the adaptive system sort of wakes up and deals in a very specific way with that particular threat. What happens, what's believed to happen with SARS-CoV-2 is that innate response is delayed to some extent. So that's the big trick that SARS-CoV-2 has, and therefore the virus begins to replicate. But for many people, that they eventually get going with the innate system, and the combination of the innate system and the adaptive system basically gets on top of the virus. And so this is what's happening to the 40% of the population that are asymptomatic, or the population, the part of the population that, let's say, have a milder infection. The people that get the severe SARS infection, the model says that basically the innate system is just really slow at getting started. The virus really gets a head start. And by the time the innate system gets going, and it then escalates, and perhaps it's too little, too late, or you might say it's too much, too late, and leads to the severe disease. Obviously, with immunization, we're trying to mimic what goes on in panel A by waking up that the T cells and the B cells. So there are the various branches of the adaptive immune system. There's the B cells producing antibodies, and then there's either CD4 or CD8 T cells. And again, this publication I'm talking about suggests that there's a much stronger correlation between a good, strong, robust CD4 T cell response and a positive outcome. But nonetheless, we are all looking to be able to help this process by administering antibodies, particularly to people that don't respond well to vaccines or people who are already infected. There have been a few reports out already from antibodies coming through. Some of those have actually suffered now because of the new variants coming through, that some of the antibodies that were originally pushed ahead are not neutralizing some of the new variants. This is a news story from last week, actually, about an antibody from GSK and Vir Biotechnology, which had a very, very nice, very good phase 3 result. So basically, it was looking at early infections and looking at people that might be likely to progress or go into hospital, and basically they identified an 85% reduction in hospitalization or death, and they're planning to fast track this one forward. The other good news about this one is it actually maintains binding against some of the circulating variants. So this antibody was based on one called S309, and I'll say more about that later on. But this originated, I think, 2003 from a SARS-CoV-1 patient, and was found from the memory response of that patient, and was found to cross-react with SARS-CoV-2, and so was fast-tracked. And I think it's that combination of binding to the to conserved regions of the virus that is perhaps allowing it to maintain its activity against some of the newly emerging variants. But I'll come back to that later on. So as I say, we joined this by-industry coordinated consortium, starting off with the lymphocytes provided by our Alkamab. Just very briefly, I'm not going to spend time on this slide. We made libraries, we did phage display panning, we generated about 1,500 antibodies and distilled that down to a set of 155, and further pushing hard on neutralization to the Wuhan strain, came up with 21 antibodies that we finished up doing developability work on. I'm not going to say anything about developability other than these antibodies were all nicely developable. Anyway, drilling down into a little more detail, the blood samples actually came from this PAXgene system. So basically you take 3 or 4 mils of patient blood and it's immediately lysed, which I have to say I was a little skeptical about that as a source of RNA, but it turned out to be pretty good. And we were able to amplify up the variable heavy and the variable light chains from this, and we either combined the heavy and light chains from the same donor, or we took the heavy chains and combined them with a naive population. Whichever route we took, we finished up with more or less the same result. Lots and lots of phage selections that, to cut a long story short, culminated in about 1,500 antibodies as IgGs that we screened. We focused in on 254 unique antibodies, which themselves had about 123 unique VHs. And then one of the first things we did was to define whether they were blockers or not of ACE2. And we did this by expressing the RBD domain of the spike protein as a CD4 fusion and capturing it on ELISA plates. Then we come in with ACE2, a dimeric ACE2 as an FC fusion, and we can detect the binding of ACE2 to the spike protein. Then the job is to see whether our antibodies will inhibit that. And the upshot of that was about 2/3 of the antibodies were in fact ACE2 blockers, and about 1/3 were non-blockers. At the same time, Alkamab, who had provided us with the RNA samples, went ahead and did next-gen sequencing of these on an individual patient-by-patient basis, generating about 3.5 million VH sequences. They then clustered these and came up with 839,000 clusters. So a cluster is basically where you've got the same V and J gene coming together, and where the CDR3 has got no more than 1 amino acid mismatch across any 10 amino acids. So it's quite a strict definition of a cluster. Of course, these are anonymous clusters. We don't really know anything about what they bind to. But of course, now with the benefit of having identified these 123 unique VHs, we can actually interrogate this database. We can kind of illuminate the database and try and learn something about the specific response to RBD. I should say most of these RBD binders are also neutralizers. And what they were able to identify from that was that there were 89 RBD binding clusters identified in these patients. And now, since they have all the data about individual patients, we can go back and look at where things came from and learn something about the patient response. So a couple of lessons jump out. So first of all, among the 839,000 clusters, there was a mean of around 4 sequences per cluster. In comparison, the RBD binders, the mean was around 116. So really, we can see there's been some sort of clonal expansion of the RBD binders relative to the rest of the population. Again, if we look at the mutation frequency in the total set, we can see that there's about 7.6 mutations per sequence relative to the germline sequence. Whereas the RBD binders that we fished out, we're finding a much lower mutation rate, something like 2.6 mutations per sequence. On top of that, the sequences of these 89 clusters, we found about 70% of them were also represented in the IgM population. So together that tells us that really we're looking at an antibody response that's driven by naive B-cell activation. I'm telling you work that we discovered as we were going through this in the spring and summer, and I think this lesson and message has been reiterated by a number of people in the meantime. Another surprising outcome was the degree of convergence. So in other words, we're finding that different patients are coming up with the same response, and in fact about a quarter of the sequences that were RBD binders were found in at least 2 individuals. And in fact, in this table, we can see this cluster number 3. It was actually found in 14 of the 19 individuals. These ones were found in 9 different individuals within the set. And of course, you can see that those cluster sizes are quite large within the data set, over 1,000 in some cases. There's also a COV-AB database that's got antibody sequences from other studies, and we incorporated these sequences into the analysis, or at least our collaborators at Alchemab did. And again, we were able to identify antibodies from other studies that matched the sequences that we were getting here. So just to summarise that section of the talk, we'd say that this early response is largely driven by naive B-cell activation, not necessarily reactivation of memory B-cells. We've also seen this convergence between patients, which, given that the initial response is largely driven by the germline encoded section, maybe that's not such a surprise after all. But we also find, both in our study and across other studies, this degree of convergence. So at this point, we focused in on 155 of the antibodies from the set, mostly being the ACE2 blocking antibodies. And as I say, these were the dominant set, and these were also where we found the most potent neutralizers, but also 34 of the non-ACE2 blockers. We also measured the affinity by Biocore of all of these, and as expected, we had a range of affinity, which you can see in the graph here, ranging from around a monovalent affinity of 70 picomolar up to a few hundred nanomolar affinity. So I'd like to now move on to characterizing these antibodies and moving in the direction of potentially generating a drug molecule. So the first thing that we did, or our collaborators at NIBSC did, was to use this pseudo-viral neutralization assay. So here we take a lentivirus and we express the SARS-CoV-2 spike protein on the surface. And inside we've encoded a luciferase gene. We infect HEC293 cells, which are expressing the ACE2 and another co-receptor. And so, of course, when an infection event takes place, luciferase gene gets turned on and we see light from these samples. So what we're looking for is the addition of antibody to reduce the light coming from the hex cells because they've blocked the infection. So when we do this, we find very pleasingly a high proportion of neutralizers. So at this point, it's a single point assay with 20 nanomolar antibody. And we're finding about 98 of them. 2/3 of them are giving greater than 80% neutralization. And I draw your attention here to S309, which is the GSK-Vir antibody, which is less potent than many of them. We took a subset of those and actually worked out an IC50 value in a dose response curve. And again, we can see a spectrum of IC50 values. These ones here, the COV2s, are actually the AstraZeneca antibodies, again, fairly potent. You can see S309 is up here in the single-digit nanomolar territory. We have many antibodies that are superior to that, but most of those, or all of those in green, are actually blockers. Up here are our non-blockers, so they're still neutralizing, but in the single-digit nanomolar territory, just like S309. But as you heard earlier, this nonetheless has turned out to be a pretty good antibody. I mean, S309, as I say, is binding to the non-ACE2 parts, and it also potentially benefits from some changes in the FC domain that give it a longer half-life, better penetration into the lung. But I think for the future, resistance to variants, which anticipate from our own as well. So we identified many potent neutralizers. Some of these, subset 21, went on to do live virus assays. And again, we're finding a number of antibodies here that are in a similar sort of ballpark to the two AstraZeneca antibodies as shown here. Again, we found there's quite a good correlation between the results of the pseudo-viral assay and the actual live viral assay, although the pseudo-viral one is more sensitive. What we didn't find was a strong correlation with affinity and neutralization. Now that could simply be down to the epitope that the antibody is binding to. Some epitopes may be better than others, but it's also possibly down to a valency effect. Of course, these IgGs are bivalent molecules, so the question is, are we also just getting a benefit from having a bivalent molecule? As I showed you already, the virus is relatively low density for the spike protein, and you can easily imagine how a bivalent IgG could bind to different trimers at the same time. This is also a structure from this publication where they've actually shown the ACE2 protein bound to the spike protein, in this case with 3 different, each of the monomers bound to ACE2. And again, we've modeled this and we can see that this is, there's a potential for bivalent binding there as well. So bottom line is there is indeed potential for inter and intra binding to the spike. And when we do the experiment, of course, that's what we find. So shown in orange here is the monovalent form of these antibodies presented as Fabs. And here we've got IC50s are in the kind of single, double-digit nanomolar territory. When we present them as bivalent IgGs, we're now down into sub-nanomolar territory, which I think together explains why we're able to take antibodies from a naive response that might be in that kind of 1 to 100 nanomolar territory that we've come to expect from naive libraries, which combined with the bivalent presentation on a multivalent virus is perhaps the reason why we get such a quick response from the naive pool. So the top panel of 21 antibodies were further characterized. Most of them, all of this group here, are actually blockers of ACE2. These two out here are non-blockers. So although they are all blocking ACE2, they all have different kind of degrees of blocking or binning versus each other. So again, even on the ACE2 site, we have a diversity of binders to choose from in terms of epitopes. I draw your attention to ION360 here, which is one of our antibodies that's in this cluster, shared with this AstraZeneca antibody, and also this ION300, which is a non-blocker. We were actually able to solve the structure of both of those, and I'll show you that here on the next slide. So shown in orange is the receptor binding domain, and shown in yellow is the receptor binding motif that interacts with ACE2. And you can see here, as expected, iron 360 is sitting right on top of that, and you can see therefore why it competes with the ACE2 site. It gets in there and blocks it. Iron 300, on the other hand, binds the back end of the virus, or at least this is the N-terminus of the of the RBD domain, and it binds to quite a distinct epitope, and not one that we've seen so far in any of the structural databases. So we are binding to this distinct, unique epitope that appears to be conserved. I'll come to that in a moment. And again, this is what it looks like on the full trimer. So I'll turn now to talk about the emergence of variants, which again over the last few months we've heard a lot about. This is a plot. This is a publication from December, and the plot in the middle, I apologise that I've obscured the legend for the x-axis, but it's time starting at January 2020 at one end and going to January 21 at the other. And on the y-axis, we're looking at the variation from the starting sequence. And so you can see that the virus is accumulating change over time. On this sort of diagonal here. There's a bit of a gap in the middle here, which I'm guessing is that, well, it would be the summer period when there was a dip in the number of infections. And so this shows this kind of, shall we call it, gradual change in the virus. On the 20th of September last year, this point here appeared from sequencing a variant in Kent. And in one fell swoop, it just took a leap forward in terms of numbers of changes. A day later, there was another version of it sequenced in London. And in one step, this virus accumulated many changes, including 14 coding amino acid changes. And again, you can see that once it emerged, it began to replicate and began to appear more and more frequently within the population. So, of course, from that, we can also create these family trees. So here's what Darwin was musing about all those years ago around evolution. We can, of course, with our modern sequencing derived techniques, create this family tree of the virus as shown here. And again, we can see this leap, if you like, compared to what's going on with all the other variants. And there's obviously ongoing mutations going on even within that clade. So looking at the protein and at the sequence level at what's going on. The Kent variant shown in blue has got multiple changes. I'm not showing them here because I'm going to focus on the RBD domain, but within the RBD domain there's a single amino acid change, this N501Y, asparagine to tyrosine. And this same variant, the same mutation appeared in the so-called South African strain. And in the South African strain, there were a couple of other amino acid changes within the RBD. But all of those are actually sitting on top and shown in yellow, the contact residues for ACE2. So basically, this is these are variants that are binding better to the ACE2 receptor, and which are being driven in this sort of Darwinian way to dominate the populations. There's also a mutation just outside of it in the South African variant that's appeared in others as well. So we wanted to know how our antibodies were faring against these emerging variants. I would draw your attention to the homology. Rather than show you sequences, there's a bit of a broad brush thing here that's saying at the N-terminus there's something like 89% homology with SARS-CoV-1. On the C-terminus, overall, it's something like 60% homology, and if you now focus in 67% with SARS-CoV-1. And if you focus in on the residues that are making contact, it's actually only 47% conservation. So we did an ELISA to determine how our antibodies were binding. But this time I actually separated the data and grouped the ones that were blocking, which one assumes is somewhere around this region, and those which are non-blocking, which in general might consider to be at this N-terminal region. And again, the point I would make would be that the antibodies that are clustered around the ACE2 binding site tend to give more potent neutralization, and more of them are neutralizers, but they're also sitting on a part of the virus that's susceptible to change, and therefore those antibodies are susceptible to lost activity. And here's the result we got. So this is 151 antibodies from our populations. On the right-hand side, I've plotted the ACE2 non-blockers, which one might imagine maybe tending towards the conserved N-terminus of the virus. And what you can see is all of them retain binding to the South African variant, which you might, on reflection, predict. And here I've got the highlighted the IN300 that I just showed you. And the asterisk is showing that these are also neutralizers of the virus. Almost all of the ACE2 blockers were also good neutralizers, so I've not highlighted that. But you can immediately see there's a fall off in the binding to the South African strain. Approximately 50% of the antibodies retain binding to the South African strain, and one can understand now, given that those are the ACE2 site as well, these changes are occurring. And again, IN360, the other one that I showed you the structure of, retains binding to the South African strain. The other thing we can look at is the retained binding to SARS-CoV-1, and one might imagine if antibody that these will reflect conserved changes, maybe changes that the virus just can't. Conserved regions that cannot be changed without badly affecting the virus, perhaps. And what we find here is that about 50% of these non-blockers continue to bind to SARS-CoV-1. And whereas with the ACE2 blocking antibodies potentially binding the C-terminus, there's only about 10% of them that are still binding to SARS-CoV-1. So, I would say, coming back to that S309 antibody, that would be basically fall into this camp here. It's retains binding to the variants. It retains binding to SARS-CoV-1, presumably because it's binding to conserved residues. And therefore, I think there's good hope for this class of antibody, even notwithstanding the fact that they appear to be less potent at first glance. So I think we, like many people in the world, chased after the most potent neutralizers we could find to the Wuhan strain. And in the course of doing that, we led ourselves to this more variable part of the virus. So perhaps as we go forward, we may be able to use antibodies such as these, either the non-blockers, or even some of these blocking antibodies that nonetheless retain binding to the variants and to SARS-CoV-1. So I'll finish there and summarise by saying we generated a panel of antibodies. We succeeded with 18 out of the 19 patients who went in, and it seems pretty clear that most of the responses we're getting are from this naive B-cell activation response with an IgM origin and very few mutations. We focused in on this panel of 155 antibodies that fell into these two groups, the ACE2 blockers and non-blockers. Found a high hit rate and high potency for neutralization with the ACE2 blockers, but some susceptibilities to viral variants. In contrast, the non-blockers, we found retained binding to the major variants, and even as far as binding to SARS-CoV-1. It just remains then for me to thank the people who were involved in this work. So at IONTAS, it was led by Anish Karat-Vilat with significant input from Georgia Bullen and the other people that you can see on the list there. I thank Sandra Ergueta for the recent variant work, and Alex for helping me with some of the nice structural images that I've been showing you. The viral work was being done at NIBSC and LifeArc. LifeArc also contributed to the structural work along with Gareth Hall at University of Leicester. Alchemab and Jane brought us into the project, and Jake did a lot of the NGS analysis, and Abcam played a critical role in just coordinating everything and doing some additional validation of the antibodies. I should say that IONTAS, the company that I formed about 8 years ago, we became part of the Fair Journey group earlier this year. And that's why you see these other names. We now work with our colleagues in Porto as part of a much bigger operation. And more recently, we established Flow 1838 as an antigen antibody production company within the group. And in case you're wondering, 1838 is when the term protein was first coined. We've developed these antibodies as part of our effort and contribution to the global effort. At one point, it felt like we'd kind of fallen behind in the race. But I think now there is opportunity here with some of the antibodies that I've shown you today to generate therapeutics. We are looking for partners to help us with that, because at the end of the day, we are a service company, but we think there's a great opportunity, and we hope we can find a way of going forward to get these into the clinic. Thank you very much. Hi there, Jan. Hi, Jan.
Coming back. Here we are. Took us a minute to get our systems coordinated.
Jan,
okay.
were you even listening?
Oh, I went off and took a nap, John. Sorry. I will start at the top of the queue then. First question, has anyone thought about developing antibodies against ACE2 instead of the receptor binding domain of spike protein?
They certainly have. It's not an area that we've been active in. It's not something that I've really kind of followed, but certainly the idea of making ACE2 antibodies is something that people have thought about. One perhaps needs to be a little bit careful that ACE2 itself has a biological function, and so one would need to make sure you weren't neutralizing that and creating side effects. But in terms of specific details, I can't really fill you in.
Well, we've had a request, so I'm going to have to ask you to multitask. The request is for you to show the summary slide again. So if you could put the presentation on the summary slide, someone would appreciate that. Sylvia, you want to ask the next question?
Yes. To go around the glycosylation problem, did you consider the possibility of developing a nanobody-based approach for developing blocking antibodies?
Again, we historically haven't done much with nanobodies here, but of course other people have, and there have been some nice studies out there. When I say we, actually, our colleagues in Porto have worked with nanobodies, and they have also generated nanobodies. But in terms of the work that I'm presenting here at IONTAS, no, we haven't done any nanobody work, but it certainly seems like a good idea. And in fact, it's quite a nice paper from Jim Wells last week showing nanobodies having an effect on viral neutralization.
Perfect.
Another question, would you predict similar convergences if you were to compare CDR usage with non-Caucasian populations?
So I would just repeat the question, please.
Sure. Would you predict similar convergences if you were to compare CDR usage with non-Caucasian populations?
I think one could probably answer that by actually looking. Let me just see. I'm not sure whether there's a time delay on here. Let me just. I guess. And this table here shows a lot of the convergence, and it actually identifies the V genes and the J genes that were coming together. So I think it boils down to a question of what V genes and J genes are available within different populations. I'm pretty sure, however, that we found matches with the AstraZeneca antibody, or some of the antibodies, and I know that some of those originated in China. So I think the answer is that we probably have already observed convergence from non-Caucasian, but I can't swear to that.
Good. So have you also found any neutralizing antibody that bind to NTD or the other domains on the spike?
So for our selection, we selected either on the receptor binding domain or on the S1 domain, which is the kind of internal part of that. So we didn't actually do an extensive search. In many ways, this was kind of under the lash, this project. Now we're kind of playing catch up and trying to work with what we had. So in an ideal world, perhaps we would have gone for full spike protein, but what we had available was a receptor binding domain or the S1 domain. But what I can say is when we selected on the S1 domain, most of the antibodies we got out were to the RBD domain anyway, and I think others have found that too.
Next question. Have you explored whether there is synergy in neutralization potency when combining multiple, now I have parentheses here, non-cross-blocking, that is multiple clones, and would an orthogonal, oligoclonal therapeutic be more resistant to virus escape from mutants?
Well, that was actually the logic behind the whole of that binning study that I told you about, was to find antibodies to different epitopes, to consider resistance by having oligoclonal mixes, and we did also look for synergy. I think there is a sniff that we may have seen synergy in a few of them, but again, that was fairly preliminary work. But there is to look for synergy, there's a lot of kind of complicated maths goes on behind it. So I'm a bit reluctant to shout out that we saw synergy because I didn't completely follow the maths on that one. But we did analyse the outcome, because you're mixing lots of different combinations of lots of different antibodies. All of them are pretty potent to start with, so you're looking for sort of marginal effects on synergy. So the short answer is maybe. and I think the oligoclonal approach is obviously a sensible approach, and it's what was behind certainly the AstraZeneca approach, and was driving the thinking both within IONTAS and within the wider consortium.
Good. So next question, can you comment on whether there was a particular phage selection technique that yielded more potent neutralizing antibodies?
I think maybe some of the people in the lab might be able to answer that question. I'm afraid my knowledge of the detail doesn't go down, but I didn't hear anyone jumping up and down and saying this is the way to go. So I suspect we could probably have got these antibodies from all of the ways we do it. We do panning, we do biotinylation, we do different rounds of selection. I suspect we got what we needed from all of them. But and end up, I think there was a huge number of selections that went on. I'm just looking here to see where was it. I think I took it out because I didn't. Oh no, I was going to say I took it out because I didn't want to show off, but now I'm going to show off. 160 different selections were done.
Okay, very good.
Next question. What do you think is the best strategy for combining antibodies, for example, neutralizing plus neutralizing, or neutralizing plus non-neutralizing, perhaps separate antibodies, combinations, bispecific antibodies? What formats are you considering?
Well, you could say all of the above, but as I say, from our point of view, to some extent, we've run out of road, in that this is not what our company is about, unfortunately, and we've kind of have to make a living by generating antibodies for clients. But all of that said, I would love to try some or all of the things that you've just listed there, and I do believe that we'll get better results from that. Again, just to pick up on the Jim Wells paper that I mentioned, again, he's finding making bispecifics has enhanced the potency of some of the input antibodies he was working with.
Another question that has to do with FC engineering. So, of course, as you know, I do this tedious and time-consuming thing where I track all kinds of antibodies. And when COVID-19 came along, and there was just this flurry of activity, which of course you participated in, various companies making antibodies for targeting SARS-CoV-2. It was interesting to see different companies take these different pathways, and one of them was differences in FC engineering. Some organizations ablated the effector functions. So there's reduction in effector functions, but different companies took different approaches. So AstraZeneca, I believe their mixture has reduced FC function, but the Vir GSK version, their antibody has increased effector function. In your opinion, after many, many years of working in this space, as applied to this particular problem, what do you think about the difference in the FC engineering approaches?
So I first of all would declare, despite my pretense here, I'm no expert on infectious diseases here, but and obviously for a lot of the historical work we do, we're looking for silent antibodies as neutralizers and so on. I've taken part in various discussions around this very topic, and I think certainly within the groups I've been speaking to, that people were inclined more to just focus on neutralization only, worried about bystander effects, let's say, from cell killing and so on. And it's perhaps a safer road, perhaps to follow, and it's obviously the route that AstraZeneca, GSK took. I think on our first cycle through this within the consortium, there was probably a general inclination to go for the sort of FC silent versions of it. However, looking at the GSK data, it's pretty impressive. The trial was stopped because it was so successful, and now they're kind of going for fast track. And there they have got functional FC complement activation, FC receptor engagement, and so on. And as I say, there's also a couple of mutations in there beyond that to increase half-life and arguably increase penetration into the lung. So I think just based on that alone, the needle probably swings more in that direction for me now. So there you're getting both neutralization and killing,
I have another question, if I can impose again on your very extensive experience working in antibody discovery and development, and it's really around the very obvious observation that the whole process was speeded up so much, whereas a year or so ago, no one was really certainly did not have an antibody therapeutic for SARS-CoV-2 in hand. And yet a year later, we have multiple of them already having achieved emergency use authorization, still some in phase 3 and some in early stage clinical studies. So what, in your opinion, is a technology or an approach or a pathway that really facilitated that speed? What helped the most, in your opinion?
Well, certainly the motivation was there for sure, wasn't it? I think as far as antibody discovery goes, we've been, doing, well, I don't know when to start my clock, but from the sort of phage display point of view, the clock started 30 years ago. And people have just got better and better at doing that sort of stuff. People have next-gen sequencing approaches, the transgenic animal approaches. I think people have, number one, people have just got better at this than we were, a decade ago or more. But there's never been a motivation like the motivation we have now, the effect it's had in the world. I think we've also seen collaboration in a way that you would never see before, sharing of data, rival companies working together. I mean, I mentioned that we became part of the Third Journey group, but at the beginning of this process, we were not. We were having a dance together, but we were rival companies, but we also got in together to work together to generate antibodies. So I think it was just that urgency and the impact it was having in the wider world. And for the guys here in the company, they were working long days, or they were staying after they'd finished their day job, because, everybody wanted to make a difference or make some contribution to it. So, so now I'm going to get them to do that for every project.
Yeah, that makes good sense, I have to say. Sylvia, did you have a question?
Yeah, can I ask a question? Since you found out that most of the antibodies were from naive cells, IgMs, do you think there would be much difference using, instead of starting from convalescent blood, starting from a naive library?
Again, others have done that, and the sort of affinities we expect to come from our naive library. So here in IONTAS, we work with a naive library of around 40 billion members. We expect to be able to take any antigen to it and to generate antibodies, majority falling between 1 and 100 nanomolar, also sub-nanomolar antibodies coming out of that. So I would have expected at the outset to be able to generate antibodies from naive libraries. And in fact, we can see from the neutralization that we're, especially in the bivalent format, that those are good neutralizers. So yes, I think you would. And in fact, there have been publications on that one. I can't remember them all. I think UMAB probably had an naive antibody library paper. In fact, was that not the origin of the antibody in the talk last time? I think so.
Yeah.
Yes is the short answer.
Good. Thank you.
Anything else, Sylvia?
So we have another question here. Have you tried IONTAS library targeting SARS-CoV-2?
Actually, I'm just looking at your questions there. Louise, thank you for yours. That was a nice question. Pedro, thank you for your Pedro Perea. I'm too modest to read that. Anyway, so what was the question? Sorry, Sylvia, could you repeat the question?
Yeah, I was just reading here the question. Have you tried IONTAS library targeting SARS-CoV-2? But
we didn't. So in this case, we just jumped straight in because we had access to the patient-derived stuff. So, my answer says yes, I would expect to get antibodies. But in this case, we had the opportunity to take potentially an immune response, although, of course, granted that most of our donors were very early, within, I think, an average of about 9 or 10 days post-diagnosis. So, but anyway, we didn't do it. It was just one thing that didn't get done.
So I'll read what John was skipping over. It says, thank you, John, you rock. There's a little bit there, John, but you do rock. That's great.
Thank you.
What was the disease severity of the 18 selected patients? Did you select those patients according to disease severity, severe, asymptomatic?
They were all hospitalized patients, and at the time that the samples were taken, they were either recovering or not recovering or severe and didn't recover at all. And in the publication, we've actually got a plot, which I haven't shown here today, looking at the frequency of the different clusters. And it's quite striking, actually. The patient who died didn't have any antibodies from the cluster. The ones who were over the worst of it had a high proportion of matching antibodies, and the ones that were still suffering were somewhere in the middle. And I've seen other publications since that say the same thing. But anyway, they were basically hospitalized patients at whatever stage of the infection that they were at the time the blood was taken.
I have another question, sorry. Do you expect, if you would take the blood from a convalescent patient from one of these new variants, like a South African, would you expect much difference in the antibody repertoire? And so differences in these antibodies you might get?
Yeah, good question. I think if that's the challenge the patient is being faced with, I suspect that they will magic up something that will work against that particular strain. But I can't say. I don't know for sure. Again, I'd be surprised if there's not somebody already doing it. Maybe somebody's already done it and published it. Everything's happening at breakneck speed. But yeah, I would have assumed that if there's a few amino acid changes in a virus, that the immune system will work out a solution.
Yeah, would be interesting.
Well, actually, come to think of it, I've just shown you panels of antibodies that bind to the South African variant, although those patients never saw the South African variant.
Sure. Yeah. Thank you.
No more questions from you, Sylvia?
Nope.
We do have a correction. So Stefan Dubel corrects us and says the answer to the question about UMAB is no. No, it wasn't. Thank you for the correction, Stefan.
Anyway, Stefan, at least I gave UMAB a plug there, you know. So.
Well, with that, I think we have concluded our question and answer period. So in concluding, I definitely want to thank Dr. Mccafferty for relating these fascinating results, and I certainly wish IONTAS and Fair Journey much success in the future. I also want to thank our attendees for joining the webinar today. An on-demand version will be available in a day or so. I'll send a link to it via email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient for you. Thanks again, and have a great rest of your day.
Thank you.
Thank
Bye.
you. Bye now.
Bye.